Hi dear students, a warm good morning to all of you. Welcome to one more session of microbiology. And today we're going to talk about the cell envelope layers outside the cell wall. We do know that the bacteria does have a cell membrane, a cell wall, and sometimes in certain bacteria we can see that there would be an outer membrane attached to it, which is also a part of the cell wall. Now, outside of this cell wall, there are certain other layers which help bacteria to live as well as contribute to its sustenance. And we're going to learn about them today. The first one, which is we need to talk about, is a capsule. The capsules are composed of polysaccharides and sometimes it will be constructed of other materials also. You can see that not all bacteria does have a capsule. Some bacteria have capsules, some do not have capsules. Now, in the case of Bacillus anthracis, you can see that the capsule is being made up of proteinaceous nature, that is, which is composing of various components such as the poly-D-glutamic acid and all that. So you cannot exclusively say that always the capsule is made up of polysaccharide. It could either be polysaccharide or be found to be proteinaceous in nature. And how can we view the capsules? We have already learned it by either by light microscopy when we use the negative staining or by using other specific capsule staining methods also. We can see the capsules. And electron microscopy, they also enables us to view a capsule. Now, when you consider a capsule, it is not required for the growth or reproduction in laboratory cultures. That is, when you're growing it in a lab, the capsule does not have much significance for the bacteria. But in the normal habitats or in its natural environments, the presence of a capsule will contribute to various advantages to the bacteria. Now, for example, if you have a pathogenic bacteria, uh, the mode by which it is being killed within our body is by phagocytosis, where phagocytes will host phagocytics, that is, those cells which can engulf the bacteria and kill it. They are referred to as a phagocytic cells. Now, in our blood, various WBC cells can carry out the process of phagocytosis to kill the pathogenic bacteria. So, if a bacteria has a capsule, it is able to overcome or resist phagocytosis when it enters into the human body. And in the natural environment, if a capsule is being present for a bacteria, it will protect the capsule against desiccation because the capsule will contain a great deal of water, or it will store a large amount of water, which will prevent the bacteria from desiccation or drying. And another factor is that various viruses as well as toxic materials such as detergents can be excluded out, or it could be protected from such materials with the help of a capsule. Now, how does a capsule look? This is a picture of Klebsiella pneumonia with its capsule. You can see, and outside of the cell there is a halo-like thing that is referred to as a capsule. The structure of capsules were mainly been the presence or the significance of capsule in pathogenicity is being explained by this experiment. In this experiment, you can see that this is called the Griffins experiment. And what is this? Let's see. In the case of Streptococcus pneumonia, they have mainly two different types of strains. One is the smooth strain and one is called the rough strain. So, in the smooth strain, they do have a capsule, and when they were being injected into a mouse, it was being found that the mouse was being killed. Whereas when the rough strain was being inoculated into the mouse, you can see that the mouse is not being harmed. That is, when the capsuleless or the R strain or the rough strain was being inoculated, the mouse was not being killed. So this indicates that capsules do have a role in the pathogenicity of Streptococcus pneumonia. Now, further on, uh, they wanted to find out what is the thing responsible for it, or what is causing the mouse to get lysed. So in this experiment, what he did is he heat killed the smooth strain and then inoculated into the mouse, and he found that the mouse remained unharmed. And further on, when the heat killed S strain and the live R strain was being mixed together and then injected, he saw that the mouse was being killed. So it indicated that uh, the something from the heat killed S strain was being transferred to the live R strain, and that was the one which was responsible for the what the pathogenicity of the organism which we have over here. So I'm not going to the I'm not going into the details of what was transferred and all that in this class, but you do note that the presence of the capsule is the one which was responsible for the pathogenicity of that particular bacteria which we are talking here, that is Streptococcus pneumonia. And the presence of the capsule was enabling it to form a smooth colony, and it is the capsule which is responsible for the pathogenicity. So now let's move on to the next type of structure which we can have. The second one is a slime layer. A slime layer is a zone of diffuse unorganized material which is being present outside of the cell wall, and it can be removed easily. Now, in the case of capsule, it has been found to be thick and all that. Means it has not been easily removable. But in the case of slime layer, it has been found to be easily removable, and it is highly diffused and unorganized material which has been found outside the cell wall. And it is mainly composed of polysaccharides, and it is not easily observed under the light microscope. In the case of gliding bacteria, they often provide slime, and in some cases, they can allow in the motility of the organism. So that's about the slime. Slime is a type of sticky type of material which is mainly being found within the bacteria. Now, the next one which we have is a bacterial glycocalyx. Okay. Now, what is glycocalyx? Glycocalyx, it mainly refers to a layer consisting of a network of polysaccharides which will extend outside from the surface of the cell. And the term can encompass both capsules and slime layer, but they are usually composed of polysaccharides. And glycocalyx aids in the attachment to the solid surfaces, and including the tissue surfaces and plant as well as the animal host. So together, the term of capsules and climbs, slime layer could be together called as glycocalyxes, and they mainly help in the attachment of bacteria to the surfaces. Like it might be to the intestinal tissues or different body surfaces. The glycocalyx will enable the bacteria to attach on. Now then we have the flagella. Now flagella is mainly being found outside the cell wall, and it is made up of chains of protein called flagellin. And attached to there is also it is anchored to the cell as well as the membrane by the basal body, and there is attached to this there is also a protein hook also is being present. Now, if you go to take the structure of the flagella is usually being found extending outside of the bacterial cell. Okay, and it is having a what does it means? It is connected. The filament is usually connected to a hook, which is in turn connected to a particular attached to a rod. Now, if you take this as a cell membrane, see you have the outer membrane, peptidoglycan layer, and the thing attached to that we do have the structure called the basal body. Okay, and the basal body of the flagellum consists of a rod and a series of rings which will anchor the flagellum to the cell wall and the cytoplasmic membrane. In the case of gram-negative bacteria, the L ring, it anchors the flagellum to the lipopolysaccharide layer of the outer membrane, while the P ring will anchor the flagellum to the peptidoglycan layer, and the MS ring, which is located in the cytoplasmic membrane, and the C ring is being present in the cytoplasm. And you can see that the MOT proteins surround the MS and the C rings of the motor and function to generate a torque for the rotation of the flagellum. So if you go to look into the flagellum, the flagellum is extending outside of the cell, and the structure of it mainly consists of a filament, a hook, and which has been attached to the basal body. And it is the basal body which is enclosed within the cell membrane and the outer membrane, peptidoglycan layer, and all these layers. The portions of these have been found. See, it consists of the L ring, P ring, the MS ring, as well as the C ring, and various proteins are also being found to be attached to it. And these proteins, called the MOT proteins, which have been surrounding this MS and the C ring, will generate a torque. Torque means the force with which it can rotate around. Okay, and you can see that different bacteria have different types of flagellar arrangement. Sometimes it might have a single flagella, and that is called as monotrichous. And sometimes it can have a tuft of flagella at one end, that is refers to as lophotrichous. And sometimes you can see flagella, okay, that is on both the sides, at both the ends of the bacteria, that is referred to as a amphitrichous. Or sometimes you can find throughout the cell the flagella, or the entire surface of the bacteria is being surrounded by flagella, that is referred to as the peritrichous nature. So based upon this flagellar arrangement, there are different terms, and different bacteria have different types of flagellar arrangement. So what does this flagella do? The flagella, these are organelles for the locomotion of the bacteria. That is, for the mobility or the motility of the bacteria do have flagella. And this flagella, they rotate both clockwise and as well as counterclockwise. That is, with the help of this, the bacteria can move either in the clockwise direction or in the counterclockwise direction. And you can see that a protein switch in the molecular motor of the basal body controls the rotation. The proteins which have been associated with the flagella will control this movement. And when they move clockwise, when the flagella moves clockwise, what happens? A tumbling motion happens within the bacteria, and it changes the direction of the bacterial movement. And on the other hand, when the flagella moves counterclockwise, it will lead to a long, straight or curved runs without a change in the direction. Let's move on. Other than the flagella, we do also have Fimbriae and Pilae, which has been found outside of the bacteria. Now, Fimbriae, they allow the attachment, and they are found in virtually all gram-negative bacteria, but not in the gram-positive bacteria. And mainly you can have two types of Pilae. Now, you have the short attachment Pilae, also called the Fimbriae, and they are usually numerous in nature. And sometimes you also have the long conjugation pilae, which are called the F or the sex pilae, and they are very few in number. Now, next we have the S layers. Now, what is the S layers? Okay. S layers, they mainly occur as a regular structure on the surface of some bacteria. And you can see that when you look on to some bacteria, they have a pattern of something like a floor tile, which is composed of protein and glycoprotein. And it has been found outer on the surface of the bacteria. Like it's like a bacteria having a, a tiled-like, floor tile-like experience. That is referred to as the S layer. In the case of gram-negative bacteria, the S layer adheres directly to the outer membrane, and in the case of gram-positive bacteria, it has mainly been associated with the outer surface of the peptidoglycan layer. Now, what are the advantages of these S layer? Uh, they do have some biological roles by protecting the cell against pH fluctuations, osmotic stress, enzymes, as well as predatory bacteria. And they also help to maintain the shape and envelope as well as the rigidity of the cells, and also promote the bacteria to adhere onto surface. Adhere means all attach onto surfaces, and in some cases it will also protect the bacteria against host defenses like by contributing towards its virulence. That is, within when a bacteria happens to enter into our body, we always try to kill it by using a host defense mechanism. So if a bacteria is having a S layer, it usually protects it from these host defenses, and that would contribute towards its virulence. And you can also see that S layer also play an important role in nanotechnology, because some of the building blocks of these S layer uh, have been used as drug delivery systems and novel detection techniques for toxic chemical bioterrorism agents and all. We can use these S layers. And the S layer proteins also have a property to self-assemble, and hence they can be used as in nanotechnology as drug delivery agents and all that. Now, how does it look? This is the structure. Like outside of the, this is an electron microgram of the S layer of Dinococcus. So you can see that outside of it, a flow tile-like appearance is being found in the bacteria, mainly because of the presence of this S layer. Now, next thing which we have to talk about is the axial filaments. As we had noticed, some bacteria are there called the spirochetes. Spirochetes are the ones which are having a type of gliding motility, and you can see that inside of them they have bundles of fibrils that will arise at the ends of the cell. That is, within the cell itself there are certain spiral like structures, and these structures are called the axial filaments, and they will enable the bacteria to have a gliding motility. And these are the external structures. In the next class we will be talking about the, now this is the axial filament. You can see this is the spirochete, and you can see this inside within the spirochete itself there is a what, some structures. These are called the axial filament, and they will enable the gliding motility. No, now, so long we were talking about the structures which have been found outside of the cell wall and all that. Now, in the next class, we will be talking about the bacterial internal structures. So, thank you for now. We will continue with the bacterial internal structures in the next session.